And he has a bachelor's in microbiology from Oregon State, a master's in microbiology from Wichita State, and then a doctorate in biochemistry and molecular biology from Oregon Health Sciences University. So I just want to point that out because if you don't graduate here from with a biochemistry degree, it's not too late for you. Still doing graduate school. He did postdoctoral work in vaccinology at the Portland VA and has had positions in both academics and private industry. He's been at Boise State on the faculty since 2004. And in case you didn't know, he works in very close collaboration with John Thurston up in the chemistry department on INRI program and now NIH programs. And he has had a lot of C of I students in his lab in the summer and after graduation. How many do you think you've had from here?
From C of I? Oh gosh, at least 12 to 20.
Yeah, that's what I thought. Yeah, ton of them. So anyway.
I know there's a point at which you just kind of lose count.
Yeah.
Like right now, my lab's a pretty big lab, and I have at least 20 students, but let's not quibble, there could be 30, you know. So what I'm going to talk to you about today is a project that relates to the work that John Thurston and I are doing in developing new antibiotics. When you're trying to develop new antibiotics, you want to identify a unique target. You certainly want to identify a target that isn't present in humans, because there's no point in developing a drug to kill an infectious agent if you also kill the human at the same time. Although the net effect is really the same, but so we're tending to look for unique targets, and then a lot of times, and you see this a lot in drug development, is people understand that a drug works. And there are certainly huge screening programs, and a lot of the basic programs like the development of penicillin and ampicillin and tetracycline, they just screened a lot of things and things worked, but they didn't necessarily know how they worked. And so what we're trying to do as part of our rational drug development is not only figure out what drugs work and what targets are good, but also why, when you inhibit a particular enzyme, does it work? And so that's led us to a bunch of kind of big studies that are kind of big data studies where we do analysis of the proteome and analysis of all of the metabolites that you find in particular organisms when you've either treated with the drug that you're interested in or when you've genetically deleted the target for the drug. And the result of this tells us shifts in the organism. And that should tell us something about how does the drug work. We know that the drugs work, but now we really want to know how they work. I'm going to say MTN a lot because it is the name of the enzyme that we're interested in. This is a picture of it here.
One in the
Oh, there we go.
Yeah.
So this is a representation of the crystallographic structure here. It's a dimer, and it's got a couple of the nucleosides here. And I'll say I'm calling it MTN because its real name is 5'-methylthioadenosine S-adenosyl homocysteine 5-deoxyadenosine nucleosidase. And so by the time I've said that, the time is up and we're all out the door. So we'll call it MTN from here on out. It's a homodimer. Depending on the species of microbe that you're looking at, it's between 23 and 35 kilodaltons. It is found just in microbes. We also find it in some plants, but importantly, we don't find it in humans. And so we've been working on this as a novel target for antibiotics for quite a while. It is the topic of a recent NIH grant to Dr. Thurston and myself because this particular enzyme also appears to be quite important in Giardia. And so Giardia is a protozoan parasite, not a bacteria. And an interesting thing that Giardia is an example of reductive evolution. It's throwing genes off right and left, but for this particular enzyme, it has duplicated the gene. So the enzyme itself must play a critical role there. So why this? Fundamentally, what we're interested in this because we're facing this huge crisis. If we were to look at drug resistance, we find since 1980 this steady rise in methicillin-resistant Staph aureus, so MRSA. Since the early 90s, we've had this rise in vancomycin-resistant Enterococcus and fluoroquinolone-resistant Pseudomonads. You may have heard of some of the more recent superbugs. There are the metallopenicillinases or beta-lactamases, and so it's become a huge problem. We are rapidly entering into what would be called the post-antibiotic era, which means that there will be infections that arise for which none of our antibiotics work. At the same time that we're facing this sort of critical shortage of new antibiotics, we're also looking at industrially a narrowing of the pipeline. The heyday of antibiotic development was actually in the 1960s. By the 1980s, the early 1980s, we were really looking at fewer than 20 new antimicrobials that were coming online. But if we look out here, 2010 to 2012, we really only had one new antimicrobial come online commercially. And the problem is, as antibiotic resistance rises, that means any antibiotic you develop only gets to be used clinically for a fairly short window of time. That means if you're going to spend $500 million to $1 billion developing that antibiotic, it better be a good one, and you better get your profit back, right? Because these are private companies that are developing these things, and they are responsible to shareholders. And shareholders, for all of their goodwill, don't want to spend their retirement fund and throw it away on an antibiotic that isn't going to end up making a profit. So much of what we try and do then in the academic sector is to try and develop new therapies. We can do these things with government support. We are trying to avoid stimulating resistance, so we'd like to develop antibiotics that aren't going to have a strong selective pressure. That means the antibiotics we develop may not necessarily kill the microbes, but they may disable things like microbial virulence. And then one of the things that we always try and do is we always try and take whatever antibiotic we're developing, we try and add it to known antibiotics, particularly known antibiotics for which there is resistance, and hoping that in combination we will drive the resistance back into sensitivity. So if we looked in my own lab here, one of the organisms we study here is a Giardia intestinalis. You can see it looks like a horseshoe crab. We also look at Borrelia burgdorferi, which causes Lyme disease, which really isn't too bad of a problem in Idaho, but has recently emerged as one of the premier infectious diseases in the US, because we've got about 300,000 cases per year if you live back on the East Coast or in the Midwestern states around the Great Lakes. We also look at a few other parasites, Entamoeba histolytica. We look at Trichomonas vaginalis. We look at Staph aureus. We look at, probably on the order of 10 different organisms. Usually I save this slide for the very end, but I kind of decided that I needed some sort of a positive reinforcement early on. So we do all of this work. It's never helpful to spend years on something only to find out that it doesn't do anything. So we have had some success. Like I said earlier, John Thurston and I have an NIH grant to develop new antimicrobials. In this particular case, we were treating Borrelia burgdorferi with some of the antimicrobials that target specifically the nucleosidase. And what we have here is an IC50 that is down around 0.3 or so micrograms per mil. The important part about this is that this particular drug works better than ampicillin does against this microbe. And so we're developing drugs that are working in the same range or better than, let's say, the competing antibiotics that are out there that are already available, and for which there's been some drug resistance developed. All right, so back to the title. I'm interested in the proteomic and the metabolomic consequences of inhibiting MTN activity. The trouble with trying to set up proteomic and metabolomic studies by taking a drug and treating cells with it is that you have to keep adding drug for a fairly long period of time before you start seeing significant shifts in either the metabolites or the proteome. So one way to approach this is to simply say, well, what is the same as 100% pharmacological blockade of an enzyme? Well, the same thing that you can do there, you can accomplish by going into the genome and deleting the gene that encodes that enzyme. And so that's ultimately what we did. We went into E. coli, we deleted the gene that encoded the nucleosidase, and said, well, this is as best as we can represent what would happen if we could always inhibit that particular enzyme activity. The only caveat to doing proteomic and metabolomic consequence type work really is that you only get a small portion of the total. So there's always the opportunity that you have missed things. So we get to say a lot of things about the things that we can find, but we can't say a lot of things about the things that we don't see. And so we certainly see that with the proteins and the metabolites, that we get only a small fraction of them, and from there we try and knit together a story about how antibiotics targeting this nucleosidase would work. So let's start by maybe a little bit of background on how some of this works. In proteomics, so proteomics is the study of the total number of proteins that an organism produces. And for an E. coli cell, that's somewhere between 3,000 and 5,000 proteins, depending on the strain that you're looking at. How you're going to identify all of those proteins, there are a lot of ways that maybe you guys have looked at already in biochemistry lab. You maybe run an SDS page gel and you've seen band patterns. SDS page gels are not super resolving, so you can usually see in one particular lane a few hundred maybe, but not in the thousands. And so what we use is a mass spectrometry approach. And how this works is we grow our E. coli, and you can see this looks like a very typical growth curve. The organisms go through a lag phase, then they go through a log phase where their doubling time is about 20 minutes. And then they reach a saturation point or a stationary phase. And so in all of our experiments, we shot for looking at samples that represented mid-log phase when we know that carbon metabolism from glucose is really active. And then we looked at stationary phase when most of the glucose in the media is gone. And now the organisms have to adapt over. They're no longer actively growing, but what they are doing is turning on a lot of secondary metabolism. So those are the two stages that we harvested from. And then again, we had condition 1, which would be a wild type E. coli. And then in condition 2, we've genetically deleted the nucleosidase. And what we do in each case then is we harvest the cells, we lyse them open, and we collect all of the soluble proteins. And again, that's only going to be a fraction of the total proteins, but it's what we can work with. From that, we're going to proteolytically digest things, and we use trypsin for this. How many of you are in biochemistry now? All of you?
We don't have cell biology or?
Have you covered trypsin yet? It's kind of one of the things we all covered.
I think we've gotten trypsin.
Okay. So trypsin is specific for lysine and arginine residues in peptides. So wherever it sees a lysine and arginine, it's going to clip right after that. And so what we're going to do when we cleave with trypsin is we're going to generate a bunch of smaller peptides that have these unique sequences. And then for our work, we're going to take one set of these from the wild type. All of those peptides are going to get a particular mass tag chemically attached to them. And we could say a mass tag of 1, let's say. And then for the knockout strain, this other set of peptides, we're going to add another mass tag. And we'll give it a mass tag, and we'll just call it 2. It's not 2, but we'll call it 2, okay? And so the point here then is we get a set of peptides here that all have a particular chemical signature added onto them. And we get a separate set of peptides here that have a unique chemical signature added onto them. That way we get to take all of these peptides and we get to mix them together. And then we're going to put them across high pressure liquid chromatography. And then we're going to subject them to mass spectrometry. And in a mass spectrometer, you're going to ionize all the peptides, and they're going to fly into a vacuum tube, and they're going to hit a detector. And you're going to get a mass to charge ratio, and from that, you can identify the peptide sequence. All right, so this is how we're comparing our wild type to our drug-treated cells. We also are comparing different phases of growth and trying to figure out if that's going to tell us anything. And so it works a little bit like this. This is just a cartoon representation of what the mass spectra coming off is going to look like. All of our different peptides are going to have particular precursor ions with mass to charge ratios that are unique, or mostly unique, we hope. And from that, however, we really can't distinguish very well between those peptides that came from wild type and those peptides that came from our MTN knockout strain. But if we treat this again, so we ionize a second time, we do an MS-MS, what ends up happening is the attached chemical signal that we put onto all of our peptides gets knocked off. And so in this particular case, let's say our wild type all got mass tag 1, and we're going to say that was red in color. It's not really red in color, but we'll call it red. And then mass tag 2 is blue. And so when we do the MS-MS, our ratio of wild type peptides to knockout peptides should be the same as our ratio of this mass tag to this mass tag. And so we use this information now to tell us relative amounts of that particular peptide. Okay, so from all of this then, we have to work back and we have to say, okay, we're going to ID the peptide. For that, we use a couple of programs, Mascot and Sequest. And then we're going to also get the relative amounts of those peptides. And so when we did this work, comparing our wild type versus our knockout strain, with any mass spectrometry project, you always want to have lots of replicates. And sort of the minimum number of replicates is about 5. And since we have pretty tight control over our growth conditions, we get very reproducible data. So we had 5 wild type, we had 5 of our knockout strain, and we looked at 2 different growth phases, log and stationary. We identified the peptide sequences, and we also looked at the different peptide amounts. We identified a total of 1,144 peptides. That's actually pretty good. That represents something like 35 to 40% of the total proteome of that organism. But it's not 90%, it's not 95%, right? So what this really tells me is we will learn some things, but the other 65 to 60%, we still don't know. What I show over here is a Venn diagram. This is sort of an initial type of analysis. Before we even start looking at the different amounts of the different proteins, what we really want to do is ask ourselves, is our data reproducible? And in this particular case, if we were to look within our 5 replicates, 67% of the peptides that we identified were shared by all 5 replicates. So that's kind of important to us too. And that's, again, it's not 90%, but what it's telling us is 2/3 of the time, everything that we've done so far is reproducible. So then we have this huge problem. We're looking at thousands of proteins, or at least, 1,100 some odd. And how are you going to display all of that data? If I give you a list of 1,000 proteins, we're looking at something like 10 pages in an Excel spreadsheet, right? It's difficult to see everything. One of the ways that we display things is just by heat maps. where we say, using a log scale, if we're looking at one particular peptide right here, for instance, we can see this band here is red. That would tend to indicate an upregulation of peptide expression there, or green bands that might indicate downregulation. It's visually interesting, and you can see a ton of data all at once. Intellectually, it tells you almost nothing, right? Because I can't look at it and say, oh, at last, you know, DNA replication proteins are all upregulated. How exciting. It's not very useful. So a lot of times we have to take all of this data, and then this is where the labor actually comes in. The experiment takes less than 2 weeks to run. The data analysis has been going on for 1.5 years. Okay, so you have to walk your way through all of those proteins and look at each one of them and ask the question, where in metabolism are they actually being used? And so that probably brings up a good point. This is a good point to look at metabolic pathways. And here is our enzyme, MTN, and I think it's critical, right? And there are me and maybe 10 other people on the planet who are with me on that one, right? But for everybody else, this might be an offshoot of a whole bunch of other stuff. But if we looked at this molecule here, acetinosyl methionine, this is kind of one of the really critical metabolites in the cell. It ends up being this group donor for about 100 different reactions. So we can take a methyl group off of it. So if we take this methyl group right off there, we get acetinosyl homocysteine. Or if we decarboxylate it and then take the propylamine group off of up of there, we get methylthioadenosine. Or in some really amazing radical reactions that have really only been looked at for the last 15 years, we can create 5-deoxyadenosine as our byproduct, and then methionine as the other byproduct. For any one of these 3 nucleosides, then, whatever we did metabolically to SAM was a utilization of some purines and a utilization of sulfur, which is a little bit difficult to get into biological systems. So what the critical role of this enzyme is to help with salvaging those components there so that we can get methionine back out and the purine adenine. The methionine and the adenine can then be used to remake this, or we could remake some other things like ATP. There are a few other things of consequence there, but the big point here that I want to point out is all of these nucleosides here that are the substrate for the nucleosidase, if they are allowed to build up, they are product inhibitors of all the reactions that make them. And so you shut down a lot of metabolism if you shut down nucleosidase activity. So, and I'm going to focus your attention over here, these radical SAM reactions, which we really didn't know much about even 10 years ago, are responsible for a lot of reactions that make vitamins inside the cell. And those vitamins are essential for enzymatic activity that regulate things like central carbon metabolism, so things like glycolysis. And so if you can't use MTN, this builds up and it inhibits here, and then you don't make vitamins, and then your metabolism starts to shut down. So we've got methylation reactions, polyamine synthesis, and vitamin synthesis, and that's really how we're thinking that inhibitors of the nucleosidase are going to work. So we are interested then in any changes in protein expression that could explain how drugs that hit this target are exerting their antibiotic effect. And we had a few ideas going in. We knew, for instance, that if methylthioadenosine built up, it was going to inhibit polyamine synthesis. And if your cell can't make polyamines, polyamines fit in the minor groove of the DNA. They're really important for helping to neutralize the negative charge on the DNA so that you can pack the DNA into small spaces. So we figured that we would see changes in polyamine synthesis from this. We also knew that we were going to start to limit things for methionine, and most of you are aware that methionine is the first amino acid that you're going to put down in any protein. If you have limits to methionine, then a lot of translation of proteins is going to shut down. We also figured that if we shut down this pathway, methylation reactions, which also do things like govern gene expression, would be altered. So there was a lot of stuff that we knew would happen, but we kind of started with polyamines because we understood that story pretty well. So if we went back to that heat map with all that data that didn't look very decipherable, and actually started looking at the peptides and assigning them to pathways, we got to see what's considered sort of the polyamine pathway. And you can see it starts off with a few different amino acids, aspartate, glutamate, and glutamine. And from these, we make some polyamines like ornithine. Ornithine is a precursor with agmatine to form putrescine, which is kind of a critical polyamine. And then SAM, which is going to go on to make spermidine with putrescine and so on. So really, for the polyamines that are important, putrescine, spermidine, and cadaverine play really big roles in helping to neutralize the charges on DNA and regulate gene expression. And so what we saw as a whole, as we looked here and here and here at all of these different enzymes, is almost across the board, we had downregulation of the peptide expression for the enzymes that were involved in these pathways. And so that tells me then, if I develop an inhibitor or a drug that targets the nucleosidase, I'm pretty sure that what's going to happen is I will downregulate polyamine biosynthesis. And that'll be one of my major mechanisms. So we saw that kind of across the board, lots of downregulation of all of these different enzymes involved in those pathways. And then we occasionally had one that went up. And so the thing that you have to understand about most metabolic pathways is metabolic pathways are rarely always on or always off. They're on or off to varying degrees. So in general, the same thing still holds true, that we are downregulating polyamine synthesis. And then we started looking at some other things. One of the pathways that we looked at, so this may not be immediately obvious to people, but this is glucose up here, and this is glycolysis. So this would be termed central carbon metabolism. We've got an offshoot here for the pentose phosphate pathway. These are really important in producing a little bit of ATP for the cell, producing pyruvate that'll go into TCA cycle and producing a lot more ATP as a result of that. Producing reducing power from the pentose phosphate pathway, that's kind of critical for metabolism too. And in general, what we saw in the knockout strain when we looked at fold changes is that we saw a lot of that was upregulated. And that was a little curious, because seeing an upregulation in protein expression ought to tell us in general that maybe the flux through this part of the pathway should be greater. And if I were just looking at the peptide data, I probably would have said that. The glycolysis was working better in the knockout strain than in the wild type, with a few small exceptions. But it didn't exactly make sense, and it didn't exactly fit all of our data, which is to say there's a reason why we study the proteome and the metabolome. It's because sometimes one set of data can be a little misleading. And so we looked at a few other things as well. One was pyruvate disposition, to try and figure out if we could tease this out even further. Pyruvate is the product of glycolysis. And to get the most energy out of pyruvate, what you're going to do is you're going to strip a carboxyl group off of it, and then you're going to attach the acetyl group onto CoA. And then that acetyl-CoA is going to combine with oxaloacetate to make citrate, and you're going to go around this cycle. And every cycle, you're going to create a couple of CO2s. And you're going to create several NADHs, and those NADHs are going to deliver their electrons to the electron transport chain. It's this cycle that allows you to generate 30 plus ATPs per molecule of glucose. If we looked in general at our peptide results, it looked like much of this cycle, especially how pyruvate was getting disposed of here, was actually upregulated. That also was a little odd to us, but you know, sometimes you just have to say, okay, well, this is my data, now I have to delve deeper and try and make sense of it. We did see a few things that got downregulated, but not much. Take-home message then, it still looked like central carbon metabolism got upregulated, and this suggested to us that the growth of the knockout strain should be really robust. But in fact, what we found when we tried to grow the knockout strain was this. Here's the wild type strain. At between 12 and 24 hours, it's already in mid-log phase. By 24 hours, it's well into stationary phase. And here's the knockout strain. So delay, depending on the experiment, of 12 to 24 hours in its growth. That doesn't scream active central carbon metabolism. What it tells you is rather poor central carbon metabolism. And there has to be some explanation for the delay in growth. So then what you have to do is you have to say, okay, well, my proteomic data says this enzyme is upregulated and it should be really robust in activity if it is upregulated. So then you have to go in and you have to do the actual enzyme assay. And so one of the main enzymes that we focused on was pyruvate dehydrogenase activity. We saw that pyruvate here on pyruvate dehydrogenase complex, we're going to add a CoA, we're going to reduce an NAD, we're going to take off a carboxyl group, and we're going to create acetyl-CoA. And this is like a critical step to getting a lot of ATP, which is really important for rapid growth. But it is a vitamin-dependent step. So it requires thiamine and it requires lipoate or lipoamide. When we looked at the activity of the wild-type strain relative to the knockout strain, even though this enzyme was upregulated in terms of protein, in terms of its activity, it was downregulated. The activity was less than half of what we saw in the wild type, even though the amount of protein was over double. That said, okay, now we've got a problem. And so it all comes back to this here. This is a vitamin-dependent step. So you can produce all the protein you want, but if you don't have the required vitamin that makes that protein active, you don't get the activity. And so we saw when we added vitamins back into the culture to replace the thiamine and lipoamide that wasn't getting made, all of a sudden we recovered a ton of activity. So let's look back here again. This is why I wanted to focus you over here. These radical SAM reactions were responsible for making thiamine, lipoate, and biotin, and those are critical vitamins for central carbon metabolism. All right, so here's sort of where the metabolite analysis comes in. The mass spectrometry was great for getting the proteomics, and we have used it some for metabolites, but we also have a really great NMR at Boise State, and so we did a lot of nuclear magnetic resonance of cell culture supernatants. And so we could identify things like formate, we could identify ethanol, succinate, acetate, and so on, just based on their NMR spectra. And we could compare the wild type here with the knockout strain, and we could ask the question then, we saw changes in enzyme expression, do we see changes in metabolites? And when we looked at all of that data, the amazing thing here was that pyruvate was almost undetectable in the wild type strain. because pyruvate has something else done to it. It goes into the TCA cycle. On the other hand, the knockout strain had an elevated amount of pyruvate, which says that pyruvate was not getting metabolized efficiently. And if we looked at acetate, acetate was higher in the wild type than in the knockout strain. Lactate was higher in the knockout strain. Glutamate was higher in the knockout strain. And I have to confess, at the point when I was looking at this, you know, you see things as if this was a linear project, but in truth, we were going at this project with many different tentacles. And my first pass at this was scratching my head saying, I don't know what the heck is going on. But when you look back at the pathways in general, and you see that pyruvate went up, and lactate went up, and glutamate went up, and you try and follow what's happening with glucose. Glucose, the cell wants glucose to go to pyruvate. And in fact, if the cell is to survive, you must do something with pyruvate. You have to do something with it. And that's because in glycolysis, you use up NAD, and you only regenerate the NAD by metabolizing pyruvate further. So, if pyruvate is going to go to ethanol, you hit a thiamine-dependent step. If pyruvate is going to go to acetyl-CoA, you hit a thiamine and lipoamide-dependent step. If pyruvate is going to shuttle off towards glycolysis or glycine, you're going to hit another thiamine-dependent step. And if you do get acetyl-CoA made, because you've managed to get around this step, it's going to hit TCA cycle, and halfway through the TCA cycle, it's going to hit another thiamine and lipoate dependent step. So, how do we explain our knockout data? In the knockout strain, we make pyruvate, and it accumulates, and you've got to do something with it. It doesn't have a lot of good avenues. One of its avenues is to make lactate, and so it does that, and it does that at approximately 20-fold more than what you're going to find in the wild type. The other avenue it has for some of this is to go to acetyl-CoA, not a lot, but when it does go to acetyl-CoA, it could go and it could make acetate. And what we see here is that this is limited because of trying to get around this step. The acetyl-CoA, if the knockout strain does make it, can go into TCA cycle, but it's going to hit this step. Alpha-ketoglutarate is going to build up, and it's going to get shuttled off towards making glutamate. And so we see that we make more glutamate there. All right, so it's sort of an exercise in like that plumbing game, where whenever you block something off, you see the green ooze going a different direction. All right, so let me summarize this. I don't have a clock here, but we'll see. Between
6:15,
okay, so we're about where I needed to be. All right. We've done a bunch of work here, and largely what we're trying to do is we're trying to explain how drugs that we develop as antibiotics are going to work. We don't necessarily want these drugs to kill the cells, although don't get me wrong, I'm plenty happy when I do kill cells with them. But a lot of times what I would like to do is for the drugs to simply have a metabolic effect that's going to do things like decrease virulence and decrease replication rates, because those things will actually allow your own body, which has a pretty robust immune system, to fight off the infections as well. The main reason not to maybe directly try and kill cells is because the lack of selective pressure then means that you don't develop a ready resistance to the drug. And some work by another fellow back east has shown that you can treat for let's say something like 50 generations with inhibitors of the nucleosidase, and you still don't develop a resistance phenotype. much of what we've done shows that we are limiting things for polyamines, we are limiting production of purines, we are also changing central carbon metabolism. So that seems like a pretty novel and unique way for antibiotics to work. We are still always ongoing in this work, so we've got a lot of additional stuff, both with E. coli 0157 as well as other organisms. We have some work that says that we can reduce the virulence expression by about 50% when we treat E. coli with some of these inhibitors. And then one of the other things that we really need to do is we actually have to go back and repeat the studies, but this time we're going to have to break down, we're going to have to treat with some of our better inhibitors. We have actually done some of this work in Borrelia, and a lot of what we found in Borrelia burgdorferi or Lyme disease is that we're getting very similar types of metabolite and proteomic modifications that we see with the E. coli. All right, so that'll kind of wrap it up. I've got a few sort of requirements here. I always have to thank our sponsors. Our funding has come through the Idaho IMBRI program. We've also got a COBRI grant running at the BRC that I'm the associate director of. We've had money from Idaho Beef Council and the Murdoch Charitable Trust, and importantly, an R15 grant that's governing a lot of the current work. Dr. John Thurston is one of our primary collaborators on this project, and then we sort of have a list of other all-stars that have been helping out. Dr. Shin Pu is the person in charge of the mass spectrometry facility at the BRC, so he's obviously played a big role. But the people who have really played a huge role are all of the students who have worked on the project. So this is maybe 70% of the students who worked in my lab a couple summers ago. They include everything from a high school student here all the way up to a couple of graduate students here. This is a minor list of everybody involved since projects tend to go on for decades.
I see 3 CNI students
Yeah,
there.
Nisha Hunter, Miranda Tang, Lacey Wayman. So Miranda was a student on the project. Nisha was a student on the project. She started off here on the project, and then she became my technician for a couple of years. The same with Lacey. She started off as a student, and she became a technician who worked on the project. We've also had a couple of graduate students working on this, and so and that's worked out really well. Reese Knipple is now at Vanderbilt University getting his Ph.D. I think something like 8 of these people are now in medical school, and my lab tends to graduate people to medical school all the time. I think I've got 10 students in my lab this year who are going through the interview process, and about 4 of them have already been accepted. So it's been an active, busy place. All right, I'll hold your attention for 30 seconds longer. I always have to put a plug in for our master's program in chemistry. So we've got a small but pretty active master's program there in chemistry. We also have a master's and Ph.D. program in biomolecular sciences. And I've got some pamphlets up here if you're interested in either of those programs. Okay, now for questions. Sure.
Matt Lay.
Oh, Matt.
Yeah, so nice to meet you.
Yeah.
In person. I was wondering about...
And wait afterwards so we can talk a little bit about the bioinformatics stuff.
Yeah. I was wondering about how B12 or some of these vitamins act in, say, vivo. Like, can the bacteria access the vitamins that, say, the host has to sort of bypass the antibiotic treatment?
Yes.
Or how little do you have to add back in culture to...
Right. To start recapitulating the growth. Mm-hmm. Right, yeah. So we can start recapitulating the growth between 10 and 25 micromolar. so not very much. And we didn't grade it out all the way, but certainly below 10, we didn't see huge improvements either. In terms of can the bacteria access things in the host, or the Giardia, or any of the enteric parasites for that matter, they certainly can access some of it, but I think a lot of times it's maybe a mistake to think that the host is terribly vitamin rich or nutrient rich. So particularly if we look at like systemic infections, once you get into the serum, you don't really find a lot of free vitamins there. Those vitamins are pretty well locked up in protein delivery proteins that your body uses. And so there is a pretty good rationale for a lot of the bacteria then to produce the vitamins that they need. Now humans are a different story, right? I think when it comes to biotin, for instance, we don't make our own biotin, right? We rely on the E. coli and other gut organisms to make it for us. So the microbes in our gut can be maybe a little bit more wasteful than we are. Yeah. About B12 specifically, I couldn't tell you. I think, what do I know about B12? It's important for methionine synthase, right? Maybe? I don't, yeah. All right, you survived.
Actually, I don't know. So, are you gonna, two things. Test these in animal models anytime soon?
Yeah, and I cut some things out. We have done some testing in animal models, so I'll just point out on this particular slide here, see all these green things? Those are E. coli that we've labeled up with GFP, and they're binding to bovine epithelial cells there. And what we definitely see with the knockout strain is that we lose probably 90% of their adherence phenotype. And then in the E. coli models that we have for in mice, the wild type kills about 40% of the mice in about 4 or 5 days. The knockout strain really is no longer lethal to the mice. So that part has worked out pretty well. And some of that's more recent, right? That, in fact, we've had difficulty publishing some of this because people wanted to see if it actually had a true phenotype. And so now we're kind of ready to send that back out again.
And then my other question was, people, have they studied separately that if you deprive cells of particular vitamins that it upregulates those, that gene expression? Because that's a pretty interesting finding, just by itself.
Right, yeah. So people have done some of that work when it, there's a fellow who worked on this with biotin, and he was looking specifically at the radical SAM reaction that produces biotin, and saw that if you limited that cell for biotin, some of the radical SAM reactions then ended up getting, that were biotin dependent, ended up getting upregulated. And so, but nobody has actually shown the things that we're seeing with pyruvate dehydrogenase complex, alpha-ketoglutarate dehydrogenase complex, and so on, which I could say maybe is novel. To a degree, when I look at it now, I think, well, duh, of course, right? It should be upregulated. It's a little bit analogous to when you're deficient in iodine. You know, are you guys familiar with iodine deficiency? Have you ever seen somebody with a goiter, right? Their thyroid gland gets huge, right? This wasn't uncommon when I was a kid. You'd see people, it was like somebody wearing a football under half of their jaw, right? In an iodine deficiency, it's not that you're unable to make the precursor to the thyroxine, it's just in the absence of iodine, it doesn't, the hormone is not active. And so So your body responds by saying, well, looks like I need to make more of that hormone, and so it causes the tissue to expand up as it's trying to make more of the hormone, but the underlying iodine deficiency is still there, so the body is trying to make tons of the hormone, and it's not active, right? And so you get these large goiters. And then if you put somebody on iodine afterwards, so that they can make the thyroxine, the goiter eventually just shrinks back down on itself. We don't see that a lot anymore because we put iodine in tons of
Iodized
things.
salt now, so.
Iodized salt, and there's iodine now in bread and other things to try and avoid that. Great, well, thanks for coming out on a snowy night.